the amount that those genes are differentially expressed is a little bit less. So this pattern here, we hypothesize that maybe SPP1 and FABP4 could be potential risk classifiers for the severity of pulmonary fibrosis.
Yeah. So in previous episodes, we saw like an expression pattern of both FABP4 and SPP1, where like SPP1 will increase and FABP4 will decrease in both IPF and even in lung cancer. And so for that reason, we want to explore further on like besides the genes, what cell types can we see and what are their roles in the disease. So this is the agenda of our session today. Okay. So where we show you three main results. So the first one will be, we show you the key players in pulmonary fibrosis progression and mediation. And then we will identify some of the epithelial cell subtypes in the disease. And after the identification, in the third result, we show you that alveolar type cells display some potential risk factors for lung cancer. Okay, so before we go into the result, let's Julie show you like what is pulmonary fibrosis.
Yeah, so for those of you who are not familiar with the disease, pulmonary fibrosis is a disease characterized by fibrosis or overgrowth of fibrous tissue in the lung, specifically in the alveoli. So when we're looking at the healthy alveoli, the air sacs are quite large and the walls of the tissue are quite thin, making gas exchange really easy within this alveoli. However, in pulmonary fibrosis patients, you can see how the walls have thickened significantly, and this impairs gas exchange between the blood and the lung tissue. Some of the key players in the disease are epithelial cells and fibroblasts. So these are kind of known facts about pulmonary fibrosis. Okay, and to go over the data set that we'll be using in today's analysis, it comes from this data set here. Single-cell RNA sequencing reveals profibrotic roles of distinct epithelial and mesenchymal lineages in pulmonary fibrosis. So here, the authors took samples from pulmonary fibrosis patients and also from control lungs, and then they performed single-cell RNA sequencing. Okay, so first step, first step, I just wanted to establish kind of the context for this disease. So identifying more key players, so the important cell types and also the important genes as well. All right, in this first analysis, here I'm querying for two genes, this collagen gene, COL1A1. You can see when we compare these different cell types, the expression of this collagen gene is found mostly in fibroblasts. And then this MMP7 is a well-known putative biomarker for pulmonary fibrosis, and this is mainly expressed in our differentiating ciliated genes. So overall, I just wanted to show in this image that fibroblasts and epithelial cells are the key players in pulmonary fibrosis progression and mediation.
Yeah, sure.
Okay. Yeah, and so for the focus of today's analysis, I am only going to focus on the fibroblasts and epithelial cells, but not necessarily the differentiating ciliated cells.
Yeah, sure. Okay. And then,
Okay.
yeah.
Mm-hmm.
Yes, and then
So
based on like, oh, I'm sorry, just go
no,
ahead.
go. Okay. Yeah, no problem. I'll go ahead. So in this heat map here, I'm just querying for a couple more ECM related genes among these different cell types to see where these ECM related genes are expressed. And we see again the collagen gene very highly expressed within our fibroblasts, but some other ECM-related genes are also expressed in these AT1, AT2 cells, which are a subtype of epithelial cells.
Basically, we see that like the ECM related genes are mainly enriched in the fibroblasts and the AT cells. And
Mhm.
which is like, I think it's quite a common knowledge, but here we just want to re-establish to make sure that, okay, so the result we find are in line with the literature.
Right. Yeah. And these figures that we produce were from bbrowser X Pro. So you're able to produce violin plots such as this one and also heat map plots like this one.
Yeah. Okay. So this is the second part of our presentation where we will we just show you that epithelial cells and then AT type 1 and type 2 are both like a common, like the main players in the disease. And now we want to go a little bit deeper into so what specific cell type in here, epithelial cell subtype that we see in the disease and their differences.
So in this first figure here, I'm looking at the epithelial cell subtype proportions in pulmonary fibrosis lungs versus our normal. And if we look at these first two categories, you can see that the AT1 and AT2 cell subtypes are at a much lower proportion in pulmonary fibrosis compared to our normal condition. And the AT stands for alveolar type. So again, these are the cells within the alveoli that's performing the gas exchange between the blood and the lung tissue.
Mhm.
And within the paper, the authors also mentioned that the role of AT2 cells as a progenitor population for AT1 cells during lung injury and repair has been well established. So, given that statement, we also kind of wanted to dive into that analysis by using pseudo-time analysis on bbrowser X Pro.
Yeah. Just a side note that like the relationship between AT2 and AT1 is also well established in literature where like the abnormal transition from AT2 to AT1 might be like an early marker for the disease. Yeah.
Yeah, so in this next portion, here I'm going to summarize how these alveolar type 1 and 2 cells display potential risk factors for lung cancer progression. So I'm going to characterize some of the gene expressions and some other analyses that we performed.
Sure.
Okay, so here I'll demonstrate this on bbrowser X Pro, how to perform a pseudo-time analysis. So switching over to our platform, to perform pseudo-time analysis, we have the tool over here on the left-hand side. You'll click it, and then you'll choose the embedding for which you want to visualize your cells on. And then you'll click Run, and it will generate a UMAP for you here. And then you can see it suggests some points and the pseudo-time trajectory for you already. So if you click on a certain centroid, then it will yield the results. But how do we know which centroid to choose? This is where you can select your metadata to see where our AT2 cells and where our AT1 cells lie on our UMAP. So that comes from the author's cell type. And again, we want to look at the transition from AT1 towards, excuse me, from AT2 towards AT1. So you can see here the AT2 cells are congregating in this top area of the UMAP. And then we see the transitional AT2 cell type down into the AT1 cell type. And when choosing a centroid or a root cell, typically we'd want to choose around an area where we see that cell state or the cell type transitioning happening. So let's say I'll choose this one here. It's kind of close to the border of our AT2 and our transitional. Okay, and then down here, it'll give you the number of cell states that the pseudo-time trajectory predicted along the pseudo-time itself. The colors here respond to the pseudo-time, but you can also change it to look at the cell states. like so. Okay, and it might take a while for the gene panel to load, but I do have the results in our slides back here. So here we see this one particular gene, FOSB, to be highly scored along this transition. So as we're transitioning from AT2 to AT1, we see an increase in the FOSB expression. And so this FOSB gene is a potential prognostic biomarker in non-small cell lung cancer, according to previous literature. And so basically, our conclusions from this analysis is that the AT2 cells in pulmonary fibrosis may transition into a population of AT1 cells that express FOSB.
Yeah, sure. Okay, so basically what we saw based on the Pseudo-time analysis is that we have a lot of AT2 and only like a fraction of AT2 transition into AT1, which align with literature because based on like many papers that we got, there is like a common knowledge that the transition is abnormal where only a fraction of AT2 become AT1. And in here, what Julie found even further is that, so the AT1 that we see here is actually like a FOSB positive or like FOSB expressed AT1 because we don't really show it here, but when we compare the FOSB expression between AT1 in the disease versus AT1 in the control, the expression of FOSB1 in the disease is like significantly higher compared to the control. Yeah.
Yeah. And so from there, the next question we want to ask is, okay, well, what other genes are enriched in our FOSB positive AT1 cell population? So here we can perform differential gene expression analysis within this population of AT1 cells, comparing our pulmonary fibrosis versus our normal. And what's interesting to see is that we see the same pattern of upregulation of SPP1 and downregulation of FABP4. But I think what is also interesting is that because we're performing this differential gene expression in this epithelial subtype population, in our past analyses, we performed differential gene expression in macrophages, and we found this pattern in macrophages. So it's interesting to see the same repeating pattern in epithelial cells.
Yeah, this is very interesting because the pattern not only appear again and again in different diseases, but also in like different cell types. And Julie, in here we have one question from the attendee is about the Pseudo-time analysis, about I think about the FOSB one, where it's asked that how the score value is computed in the Pseudo-time analysis.
Yeah, so that one we can follow up on documentation if you want to read more about the methodology. But if you're familiar with pseudotime at all, the pseudotime analysis tool on BBRGX Pro is based on Pi Monocle 3. Yeah, so we will hopefully follow up with you if you are able to provide your email for us.
Yeah, sure. Okay.
Mhm.
So, okay, in
Okay.
here, and we also have one question from Paramo. Okay, so Paramo asks, did you run pseudo-time analysis on selected cells or the whole data, both normal and IPF together?
Okay, so I ran the pseudotime analysis on just the IPF samples, and I also subclustered the population of AT1, AT2, and the transitional AT2 cells. So I knew that I wanted to look at those three cell groups together and look at the trajectory of that.
Yeah, okay. Thank you, Julie, and thank you, Paramo, for your question. Okay, so I think we can move on. Yeah.
Yeah. Okay, so from here, now that we've identified some differentially expressed genes, now we want to characterize, okay, what certain pathways are upregulated in our population of AT1 cells. And when I ran the gene set enrichment analysis, I do find in upregulation, of course, in this lung cancer pathway, the small cell lung cancer pathway.
Mhm. Yeah. So basically, this is like a small site. We can say that. So in FOSB positive AT cell, we see like an early phase of some enrichment of small cell lung cancer related pathway. Yeah.
Mhm.
Okay.
Okay. Yeah, and so from here, we can also see, well, now that we know what pathways are upregulated, we'd want to ask, okay, what cell types do our AT1 cells communicate with? And we can do that by using the cell-cell communication tool on the browser X Pro, which I can also demonstrate.
Mhm.
So over here, the tool is located on the left-hand side. We'll click that, and the cell type metadata we'll choose is the author's cell type. So here you can see we're going to compare 31 different cell types together. You can also change the number of permutations. You'll click Run, and the results page will yield a heat map that is interactive.
Okay.
All right, so here in this heat map, you can see our sender cells are on the y-axis and our receiver cells are on the x-axis. And for each of these nodes, you can hover over it and you can see the number of significant interactions as well as what the sender and receiver cell is. If we click on a particular one, so I see that here, our AT1 cells are sending a lot of signal to these KRT5 negative and KRT17 positive cells, which are another subtype of epithelial cells. So I'll click on this node. You can see the number of significant interactions is
Mhm.
96. And it opens up the table of the interaction pairs. So these are the ligand and receptor pairs, as well as the pathway that's associated with it, and the p-value and the score as well. Yeah, so taking a look at some of these pathways, we see enrichment in EGF pathways, epithelial growth factor pathways, collagen, and laminin. And then I can really quickly show you guys some other features in the cell-cell communication as well. So you can gate for the number of significant interactions or the max score by using this filter here.
Mhm.
But you can also kind of filter for the interactions by inputting specific pathways, like so. And then finally, over here in the network, it'll give you this network graph where if you hover over a specific interaction, it'll show you the number of significant interactions and the sender and receiver cells. Okay, so that covers an overview of the cell-cell communication tool on bbrowser X Pro. Going back to our results and what we found throughout this analysis. Um, So again, we see a lot of interaction between AT1 cells and this subtype of epithelial cells. And in the paper, the authors identified that these subtype of epithelial cells overlay some of the areas where there's a lot of fibrosis occurring, and they're able to verify this through IF imaging.
Julie, before we continue, I think we have
Yes.
another question from Paramo about like whether because Paramo don't see the cell-to-cell communication on the talk
Mhm.
to data window. So I just before we move on, I just want to answer that. So right now, Julie is using bbrowserx Pro, which is a newer version of bbrowserx. And for like talk to data, like exploring public data on talk to data, and then you want to use cell-to-cell communication, this feature will be available soon when you explore public data on talk to data beta version. Yes, so this is you can see like exploring on the beta version. and it will be available quite soon. We will send you an email to let you know when the feature is available. Yeah, thank you. Okay, yes, I think we can move on.
Okay. Okay, and so again, to summarize some of the pathways that were enriched within that interaction of cells, we see that AT1 cells communicate via the CCAM pathway. So you see this pathway is the highest scoring pathway, and its interaction pair is through the CCAM6 gene. And from previous literature, CCAM6 has been found to be overexpressed in lung cancer cells. So again, we see the same repeating pattern of lung cancer characteristics. For the rest of the pathways that we see enriched relating to laminin and collagen, these are correlated with overactivity of laminin and collagen pathways, which lead to the growth of fibrous tissue in our pulmonary fibrosis tissue samples. Okay. And to summarize our findings that we found today, so we know that fibroblasts play a profibrotic role in pulmonary fibrosis, while epithelial cells can act either as a mediator or a progressor of the disease. And in pulmonary fibrosis lungs, they have a lower proportion of the alveolar type cells compared to normal. We also found that the AT1 cells exhibit upregulation in lung cancer related pathways, and that these cells communicate with other epithelial cells via the CCAM pathway. And when we, and perhaps like an overall takeaway is also if we were to target these FOSB positive AT1 cell population, this could present as a potential therapeutic target. So this could definitely warrant more analysis from the findings we found here today.
Yes, and that's about the findings and the presentation that we want to show you today. And now, okay. And now, please, we have some question for you about the new platforms and also some features that we would like to ask more. So please answer the question. There are only 3 questions, and please take your time. And why you are answering the question, we also received some question from the user. So, okay, so Berman asked us to, can you please send a link to the most recent version available where I can use Pseudo-time? So Berman, my college will share with you, like we will follow up with you, the link and also the current version of bBrowserX Pro, where you can use the Pseudo-time analysis and also cell-to-cell communication analysis. Yeah. And we also have another question from anonymous attendee where the question is, can we show again on CMA3B expression on this data, like in IPFs and cancer data? So I think in this specific data set, we only have like the IPF and normal, but like we will try to show the CMA3B expression and see like what are the differences. But if you want to visualize like the differences between IPF and cancer, you can go to talk to data and then try to query this gene there. I believe it will be very quick, especially when you go to the beta version. You can like, compare directly between IPF and cancer. Yeah.
Yeah, so I can quickly show a couple different ways where we can look at the expression of CMA3B between our 2 different conditions, IPF and normal. So one way is you can simply query for CMA3B here. You'll click search, and then you can see how the expression kind of congregates in this area. If we switch the metadata to condition, the conditions aren't really separated well on the UMAP because there isn't too much like differences between, but a more, I guess, how do I say, a more specific view, or how to get a more specific view of this is to use our visualization tools. So you can build basic charts here, like our violin plot, and look at the expression of CMA3B between pulmonary fibrosis and normal. And you can switch between these different chart types here. Maybe a better way to show this is by using the composition tool here. Okay. So the expression of this is quite low, but if we hover over it, the average expression here is 1.14, and here it's 1.07. So it's very slightly higher than normal, although it's hard to say whether that difference is significant between these two conditions.
Yes. Or I think that maybe this gene expression is quite like specific to a cell type, and therefore we might need like more information on the specific cell type expressing this gene and like, oh,
Yeah, that could be as well.
and yeah, okay. So that's about this one. Okay. And now we'll move on with the next one. So the question is, not all the studies will have the cell-to-cell interaction function, right?
Yeah, so I guess there's some confusion on like how the studies are kind of appearing. So while this data set is available on Talk to Data, when you open up and explore that data set, it's going to open up the bbrowser X UI. So it will not have the cell-cell communication feature on that UI. But if you were to like import that data set into bbrowser X, that's when you'll be able to have that cell-cell communication feature.
Yeah. Okay, done. So thank you, Fernandez, for your question. And now let me just, okay. Okay, so I think we can continue with with the question from symmetry B. So in here, so Julie, I think we can add more level on, like we can add the cell type composition here. So we can see like, so what cells really express symmetry B.
Sure. So it's gonna build quite a large graph,
Yes, because
like
here we got
generate.
like 31 cell types,
Yeah.
but we can see
So
quite clear. Yeah,
yeah, in our normal here, okay, so it
expressed in
stops here.
AC1.
Yeah, very highly expressed in these AT1 cells. And then down here,
Mm-hmm.
yeah, again, expressed in AT1 cells, more highly expressed in our pulmonary fibrosis AT1
Mm-hmm.
cells.
Yeah, sure. Okay, so I hope that answered your question, and thank you for your question again. Okay, so let me just wait for another question, and then, okay, answer here. Okay. Yes. And so please, if you have any other questions, just feel free to send us. Otherwise, if you want to, book a separate session so we can explain more for you about any features or anything on our ecosystem, please feel free to use the link on the chat. And yes, okay. Yes, thank you on the question on symmetry B. Okay, thank you. So
Thank you,
yeah,
everyone.
thank you everyone. So I think that is everything for today. We will return next week. And thank you so much for your attention,
Yeah,
and see
thank you
you
for
later.
joining.
Yes, bye.